Thank you for the build-up, and I haven't spoken in front of a live audience in quite a while, so please bear with me. If you could all just put your phones up and I can picture a computer screen, that'd make it a little easier for me. But no, I'm grateful to be here and for this opportunity, and this year I'm going to be talking about from microbiome to microbiome and how the environmental microbiome that we're all working in and looking at, how this really impacts everything in our world. Next year, if you come, I'm going to talk about a Where's Waldo kind of moment, and we'll see Lost in the World with Travis Bowman, because I've done that a few times. So I'll bring some pictures for you of that. But as Travis said, my name's Dora Taggart. I'm the CEO at Microbial Insights. And today I just want to share with you a little about the amazing legacy that I've seen in my 21 years with the company and in environmental remediation as a whole. But even more than that, I really want to frame this discussion around the impact of genetics, how the environmental microbes in the subsurface of a contaminated site can prevent damage to our gut microbiome, and how these two types of microbiomes are actually quite similar when you look at the environmental and the gut microbiome. There's similarities and connections that are really important. And I hope I'm going to give you a couple of takeaways from this talk today. One, how important bioremediation is, and all of us in this room, the work that we've been doing for the last 30, 40 years. I know I've listened to some great talks about this. The work that you're doing is really key to human health. And then second, how possible it is to transition the tools that we're using in the medical industry to environmental restoration and vice versa, back and forth. And then three, the interrelationship between every aspect of our world and our health. So Sam's got some little microbe bin, and if somebody can guess this number, I'm going to have him throw something at you. I know you want to be beamed during lunch today. So how many billions, I'll give you a hint, it's billions, of chemical letters do you think it takes to make up a human being? Anybody got a guess? 72 billion? Lower. Keep going lower. A little bit lower. Keep going. Down. Up from 1. Did I hear 3 somewhere? All right, you get a microbe man. Woohoo! Right here in the front. Easy one for you, Sam. Everybody in the middle, we're really sorry. Okay, maybe Sam should have been my pitcher, okay? He has other assets for the team. Okay, so we know that we need 3 billion letters to make up a human being, chemical letters, right? But each of us is so unique and individual and unlike anyone else on the planet. And I know some of you are thinking, thank God there's not another Dora, because I've seen her picture enough this week, right? But we're all connected to every form of life on this planet. We all share the same fundamental mechanics. We all share this ever-evolving earth. And after all, that's the blessing, the beauty, and really the magic of life as a whole. The founder of Microbial Insights, Dr. David White, really understood this. He knew all the connections to our health, and especially he understood the threat that environmental contaminants really pose to our systems that are our first line of defense. Dr. White was both a medical doctor and a PhD, so he thought, how do I take these tools in medicine where they have money and put it in environmental cleanup where we have no money? And so over the 30 years that this company's been founded, it's been a blessing for me to really see all the different things that have evolved from these tools and the application. So we think about molecular tools for remediation, right? We might think about it for corrosion in other industries, but what about our human gut microbiology? I mean, qPCR has gotten a lot of recognition with the pandemic, right? We now know that. It's a household name around the world. This has gotten, I don't have to explain that to everyone anymore what qPCR is. We know how accurate and valuable it is, but how do we use these other tools? So I want to show you some really cool projects and get a little geeked out by the stuff that we've done, looking at not only environmental restoration and cleanup, but how these microbiomes are important for World Heritage and cultural sites. How it's important for wildlife habitats and actually even exhibits of wildlife. I'm going to tell you about a cool example of that. We're going to talk a little bit about the ocean and ocean floor cleanup, and then end with some work about the astrobiology. But the legacy that Dr. White left for us is why we care at Microbial Insights, and it's why we continue to pursue what we have to combat environmental threats and threats to our health. So I really want to talk about these projects and kind of share a little bit of that legacy with you today. First, I want to start with the human gut, because that's a topic I've gotten really excited about over the last few years. We've done some really cool work with the National Institute of Health and just looking at how these tools are applicable and what happens in our gut. And when we're talking about our gut microbiology, we're talking about something really complex, right? We're talking about tens or hundreds of trillions of cells that are contained in our gut microbiome, and that's equal to, if not more than, the number of human cells in our body. So the gut microbiome is critically important, and we're talking about extreme diversity when we talk about this. We're talking about 3.3 million non-redundant genes that are present in our gut. So taking care of ourselves is critical, and it shouldn't be a surprise to us that this collection of microbes that we carry around with us every day have direct effects on the chemistry that happens in our bodies, and by relation, direct effects on our health as well. So there have been a lot of studies that have shown the microbiome's utility as a predictor of health and disease, and that's what I want us to think about, because environmental restoration, we're great at using molecular tools to understand what's happened, quantify who's there, what do we have right now, but how do we start to use these things to predict what's going to happen down the road, to use it as if we can harness that power and that energy. So all these studies that have been done with the gut microbiota have showed us that the composition of our gut microbiome is actually a better indicator of our body mass, of our blood glucose levels, of our cholesterol levels, and cardiac health than any genetic parameter that we have. So the composition of the microbiota in amniotic fluid can actually predict preterm birth. You can predict with cirrhosis patients the longevity of disease, the severity of what the disease is going to be, just by looking at the microbiome of their gut. So taking all that together, the studies really suggest that the microbiota is not only our first line of defense against disease, but we can also use it as an indicator of our health and disease susceptibility. So relating this back to the environment, because that's what we're here thinking about this week, right? There was a great paper that came out by Van de Wild in 2005, and they were talking about PAHs. There was a talk this morning about a PAH-impacted site. And this paper showed that when PAHs pass through a human system, it can be converted to estrogenic compounds by our colon microbiota. So while these PAHs are known and reported to be toxic, and many of us work on them, how many of you have had a pH site that you've worked on here, right? So you're impacting not only environmental cleanup, but the research shows that the risks are still underestimated because we have biotransformation that's happening in our bodies. So what we're doing is so critical for the world. And we know that a number of toxins are modulated by our gut microbiology, right? In some cases, microbiomes can reduce toxicity. We see that in the environment. Some cases, we can increase it with mobility and changes. So there's a great review paper that I've listed on this side as well that I would highly recommend. And one of the examples in this is about the methylation of mercury that happens in our gut. So we all know that methylmercury is one of the most toxic forms of mercury. So microbiome conversion can increase toxicity, but what we found in this paper is, on the other hand, they had pregnant women in Tanzania that they gave probiotics to, and they actually saw decreased levels of mercury and arsenic in their blood. So understanding our microbiomes, harnessing that power, is really going to determine what happens to a toxin, how toxic a toxin really is for us, and what happens when we break it down in our body. And not only that, we're talking about all these great tools that are out, next generation sequencing, all the different DNA, the human genome is now being completely sequenced. We have so much depth of knowledge about sequences, and changes in diversity in a microbiome are super important. When it comes to the gut microbiome, we know this without a doubt because studies have shown that microbial diversity within mice change dramatically when they're exposed to heavy metals or toxic elements. We also know that certain chemicals and herbicides have been correlated with change in the gut microbiome of mice, and guess what? It resulted in depression and anxiety. Not sure how they measure that in mice, but we know that as a society that we're seeing changes in our mental health, right, as a population. So monitoring microbial diversity within a microbiome, either environmental or gut, can really paint a clearer picture that will specify the impacts related to that toxic contamination. And the bacteria in your gut are responsible for producing many of the neurotransmitters that play the key role in your brain, your overall mood and health. Researchers estimate that 90% of your happy hormone, serotonin, is actually induced from your gut. So the bacteria in your gut tell your body you need to produce more of that to be happier. So we need our gut to be really happy, right? We want the right guys in there. Other neurotransmitters also that regulate mood and mental health, like dopamine and norepinephrine, all of those are actually produced in our gut as well. So there still needs to be a lot of research because we don't understand that blood-brain barrier and how those interactions are all happening. So there's still a lot of work to be done. But at the end of the day, we need to understand that everything interacts with everything else. So when you guys are doing groundwater cleanup, you're impacting the agriculture, what's going to grow there, the foods that grow. Those foods are getting introduced into our guts, into our kids, into society as a whole, right? Animals are being affected. So the work that we do is really powerful. And that brings me to what I'm so proud of that we've done over the last 40 plus years in this industry is to look at the global impact. And Travis asked this morning about the history, because I'm the old lady on the panel. Trent got the future. But over the last 20 years, I have seen a big change in the uptake. At Microbial Insights, as you saw in the video, we processed over 100,000 qPCR samples from sites all over the world. So we've seen sites from every state in the US, more than 40 different countries around the world, and it continues to grow. And you know, when we started this, I remember thinking about Dihalococoides and that impact, as we talked about this morning. But now it's not just chlorinated compounds. We're talking about petroleum hydrocarbons, we're talking about PFAS, we're talking about 1,4-dioxane. Whatever the contaminant is, microbes are an important part of us understanding those processes. And not only when we're talking about bioremediation, when we're talking about monitored natural attenuation, what do you think the natural process is? It's those little guys. They're the ones that are important. If you heat a site, you have the thermal volatilization, but what happens on that boundary? Those organisms are enhanced because they get excited. They're happy with the heat in the peripheral zone. And we do chemical reduction. Anything we do, we're leaving residual products that are used by those microorganisms either in a positive or a negative way. So understanding those organisms and the power is really what helps. And we're seeing this all over the world. You know, the US is great to be a leader to start a lot of this research and the work that's been done. And we're based in Knoxville, Tennessee. We get samples from all over the world. I'm just so proud to see all the government agencies of the world that are taking into account the research that you guys are doing, the important studies that are being produced, and turning that into something that's beneficial. But to learn, to move forward for the future, we have to look at our past, right? Start to think about what did we learn, what mistakes did we make, and how do we do better in the future. So I want to share with you guys some interesting projects because this is how I get all nerded out every morning when I'm coming to work. So microbiomes are important, right? We've established that, our gut microbiology, our environmental microbiology. But what about estuaries and looking at uptake into fish? We can get a lot of metals that are in our fish, changes in those aquatic environments. We did a really cool project, and I know it's lunchtime, so I'll be not too much description on this, but the acid rain changes that are happening in our rainforest. We're changing the biome of our rainforest, right, with the environmental changes, with the things that are in our air, more acid rain production. And so we had a researcher that actually was looking at this, so he collected all the snail poop, and I cannot imagine having that job, but he went through the rainforest collecting snail poop, and he sent it to us for analysis, and we looked at how the microbiome of those snails was shifting over time with those acidic events, how that impacted not only the snail, but other animals as well. And then, you know, marine environments. So we have a really cool project, and I can't disclose the name, but think about the biggest amusement park people in the world. And they had this really cool fish exhibit. They go in one morning, it's glowing. What do? They don't want the people to come in and be freaked out by this bioluminescence that's happening. They had treated it with their normal protocol for bioluminescent bacteria. It's not bacteria. So what are they going to do? So they sent us a sample, and what we figured out, it was an algal bloom. It was a bioluminescent algal bloom that was happening, so they had to do a different treatment to control that tank. May not have had a big impact on our world, but it made a tank a lot prettier. So the other thing that we've been working on that I'm really proud of are World Heritage and cultural sites. We want to preserve these things for our children, for future generations, right? And so we don't often think about the importance of the microbiome that's on these objects. So biodeterioration, how does it happen? Microbes and fungi are the ones that are utilizing and changing these. So we've done some really cool work over the years with the Getty Institute, looking at different cultural heritage sites like the Magao grottos on the Silk Road in China. We got to look at some fungal growth that was happening there and help them understand what fungi so they could treat those without damaging the artifact. The Ames House and the Salk Institute, there's stuff growing on the outside of these buildings. How do you protect these beautiful structures? So we got to identify those microorganisms. And then the study that everyone's been waiting for, and the question of why does Dora have a tomb next to her picture in all these ads is, and Travis wanting me to import that. I'm not sure how that would work. But we did some work with King Tut's tomb. So if you're interested in this, there's actually a PBS special, and if you'll email me, I can send you a link to it. But we were involved with the Getty Institute and Princeton University. Definitely not historians, so our piece of this pie was really small. But what they had was there's fungi growing all in Tut's tomb. Is this ancient fungi, or was it the archaeologists that discovered the tomb and went in there and introduced all this fungi? Where did it come from? So that was the question, and they wanted to understand more about Tut's death, all the mystery behind Tut's death. So what the historians determined, based on some of the things that we found, and we did find aspergillus, penicillin, some of the normal fungi, but there were things that were unidentifiable that we couldn't sequence and figure out what they were. But what they found is, King Tut was a young king. He had a massive impact during his time in his reign, but he was very young. He had this big, ornate, beautiful tomb that he created for his afterlife, and it's all this paintings and this massive structure. Well, when he dies suddenly in battle, what the historians think happened is that the king that came after him was like, wait a minute, I'm going to put this kid in a big, huge tomb. I don't have such a nice place. So they think he took Tut's tomb for his resting place, and he created a smaller place for Tut. So we had to paint it really quickly and put Tut in there, close it up, because it wasn't pre-planned, right? It's a fast movement. So what happens when you put wet stuff in a dark environment? What are you going to get growing in there? Fungi, right? So that's what they think happened, and that explains King Tut having a much smaller tomb than a man of his stature typically would have had in history. But if you're interested, definitely reach out. That was a really cool project to be involved in and get to see the evolution of that. We've done a lot of work over the years with astrobiology. So we're touching not only this world, but worlds beyond. So we're working with NASA looking at Mars analog environments. So is there life on Mars? I tend to think so. Maybe it's not aliens, but there's definitely probably some bacteria and things that can survive that are created in those planets. So they've sent samples from the Mojave Desert, the Atacama Desert, Death Valley, all of these places to kind of mimic life on Mars and see what we can identify and what happens in those really extreme environments. And then as a young scientist, I can remember this. I'm in my 20s, and I'm holding lunar rocks in my hand doing extractions. And that was so powerful and inspiring to me as a young scientist in this field to think about what is happening on other planets and looking at those bacterial profiles and the power of the tools we have now. If we'd have had those back in the early 2000s when we did this experiment, I can't imagine what we'd identified. We could only identify 3 to 5 dominant microorganisms back in those days. Now we can get tens of thousands of organisms identified in our samples. So in those days, we had a few identifications that we made that we suspect came from the transit back more than from the moon surface itself. But there were some unidentified sequences, whether it's because technology was so bad in those days or if it was something really unique to that planet. We don't know the answer to that, but that's kind of where that project was. And then we've also done work with NASA on early Earth, looking at Antarctic dry valleys. How was the Earth formed? How did things evolve over time with the freeze events that happened? Looking at the Arctic permafrost, the North Dakota clays, really understanding how we got to where we are today. And then the last NASA project that I'll talk to you about that I'm really proud of the work that we did is NASA sent us samples because they had growth in their Mars chamber. This is supposed to be a sterile environment. Shouldn't have anything growing in there, right? So they sent it to us and said, what in the world's going on? What is this? So this is back when Dr. White was still alive, and some of the researchers there, predates my time a little bit, but they took those samples, identified the microorganisms that were in there, and then they actually traced it back to, this is probably the gas that's leaking into this chamber. This is where your leak is originating. And sure enough, it was, and they were able to make a resolution. So problem solving with these microbiome results is really important and sometimes overlooked. And then I read about this article, so I just had to share it with you guys because I thought it was really cool. A few years ago, they sent this microorganism on the skin of the International Space Station. And this organism lived in space for over a year, and they wanted to see what happened to it. It's Glyocapsa magma. And what they found is that it survived on the skin of the space station, which is pretty phenomenal in that extreme environment, right? So what does that tell us? It's not just wasting money to do this, right? But it told us that they live in protective clusters. How do these microorganisms live and survive through this? And not only that, but they found that these bacteria have really cool DNA repair capabilities. What does that mean to us as humans? If we can start to identify those genes, we can figure out how to do better DNA repair, we can translate that to medical field and just have an enormous impact on society. So I'm really proud of the work that has been done here and in other galaxies. It's really exciting. And as I end today on some of the cool case studies talking about the ocean, I think a lot of you in this room probably had some impact and work in the Deepwater Horizon event that happened. And I'm really proud these pictures, it's kind of hard to see. These are our biotrap samplers. And Dr. Terry Hazen's group actually deployed these in robotic devices that went tens of thousands of feet down into the ocean so that we could look at degradation of those compounds as they sank into the ocean. So it was really cool to be involved in that study. And then we got a call. There had been a spill. There was contamination all the way down to the ocean floor. So they're like, how do we sample the ocean floor? That's a pretty big request, right? So what we came up with were these piezometers, these gray tubes here, and you can see the cone shape on the end. So we put biotraps in them, and then they were loaded with different compounds of interest. So they wanted to know, are these particular long-chain hydrocarbons going to degrade over time and how quickly? So we labeled some 13 carbon, put those compounds down on a biotrap, and divers took them down and installed them and hammered them down into the ocean sediment. We left them for a period of 30 to 60 days, pulled them back, and looked at degradation rates of what was happening with those compounds over time, and proof positive that the compounds could degrade at that depth. So it was a pretty fun study. I can't tell you exactly where in the world it was, but it was off the coast, and it was a pretty substantial impact and exciting that we got to be part of that cleanup. So before I finish up, I've been thinking a lot with this talk about these molecular biological tools, the evolution, what we've seen, and I wanted to discuss what I think lies ahead. What's the future hold for us? So we know that qPCR and quantarray are accurate, right? COVID helped prove that to the world. We know that we can look for a lot of different microorganisms and genes, but what do we know about activity when we're looking at the DNA? We can look at the RNA like Trent was talking about this morning and start to get a little closer to activity. We can look at proteomics. Those things are coming down the pike, right? But they're not commercially available yet. We can do things like next generation sequencing, where I talked about that we can look at the total microbiome, what all is being detected in our samples. But what about the health? We know the genera of all these different guys that are present. What does that tell us about the health of that particular environment? And then we have tools like compound-specific isotope analysis and stable isotope probing that we can use that are great. They can help us prove contaminants are degrading. But that is what's happening currently. How do we predict the future? How do we use these tools to be more predictive? Kind of the whole point of thinking about our gut and what we're doing. What if we can do more? What if we can predict a cis-DCE stall is going to happen before it does, so we can start to manage things before they occur and be proactive rather than reactive in our environments? How do we look at the activity versus inactivity of key degraders in the subsurface and be sure to stimulate them correctly? And there's a tool that I'm pretty excited about that I wanted to share with you guys. It's called Metabolomics. It's been around for a long time, Proof of that is this 1997 paper, and this is where they analyzed the blood of over 44,000 individuals. When we're talking about metabolites, we're talking about the small molecules that are produced through biotic processes typically. And so looking at all these detectable small molecules within 40,000 people, and what they found is they tracked these people over 17 years, and they found that 5,512 of these individuals died. And Joris Deline, who was the lead researcher on this and wrote this paper, described in this 2019 publication that these metabolic profiles of the individuals acted as a better indicator of mortality than conventional factors like body mass, blood pressure, cholesterol. In fact, he took every predictor we would typically use and combined them, and Metabolomics gave him better predictive capabilities than everything else we currently do. So pretty powerful tool. The study of the small molecules in a sample, I think, is really going to be very powerful when we use it with the right statistical analysis. So we've only begun to imagine what it can look like if we apply this in the environmental field, what we can predict, what we can learn. And that's one thing that we've been working on. We have an NIH SBIR grant that we just completed with the University of Tennessee, and we've been learning how to use these metabolomics for environmental focus. So as you can imagine, the science has dramatically improved since 1997, so we can actually look at tens of thousands of molecules in a sample rather than just a few hundred. And it's important to clarify that we may not know the name of every metabolite that we detect when we get these 10,000 metabolites, right? But we're looking at the overall profile at those chromatograms, and we maybe identify 10% of them and know their function. But how do we learn more? By amassing this data, starting to understand what it means in our society. So by comparing these profiles in known and controlled situations, we can start to identify patterns that can help us predict what's going to happen in the subsurface. So what you can see here is Dr. Loeffler's group took a few different Dihalococoides strains and a Dichloromonas and looked at the metabolite profiles, doing nothing but PLSDA plots to see how they separate out. Statistical analysis of this, you can see that even the strains have very distinct metabolite profiles, right? So that was exciting. Then moving to the next step, how do they change? Do the metabolites always stay the same? Of course not. When they're inactive, they have one certain profile. As they become more active, you can see again with these PLSDA plots, and depending on what contaminant we feed it, you see changes in these profiles that can then be used to be more predictive. So we got all excited about this and submitted for this proposal, and during the proposal we looked at 3 chlorinated sites, and what we did was we wanted to understand cis-DCE stalls when they happen. Can we see differences in the metabolomic profiles of these stalls? So we got those 3 sites. We had areas of the site that had active reductive dechlorination, background samples, and areas with cis-DCE. We were able to identify over 12,000 metabolites in these samples. And if you look at the PLSDA plot again, you can see these clear groupings. Here's your background samples, here's your active reductive dechlorination, and these are the ones that stalled. So if we can screen our sites with this and use this as a predictive tool of what's going to happen, we can be much more cost effective in the implementation of what we do at a site. And not only that, we're really super stoked because we were able to identify 5 key metabolites that we think are going to be really important for reductive dechlorination. So we want to kind of move forward to a bigger field study to be able to look at this a little more. So I hope that my talk today shows you just how impressive bacteria are. All microorganisms really get me all excited and make me happy. But also the tools that we use to analyze these microorganisms are critically important and can help us really understand the relationship, the interdisciplinary, to steal Trent's word, actions that are happening in our world as a whole, and to be able to look at that and how much the work that we're doing and will continue to do impacts not only the location where you're doing the cleanup, but you're impacting everything that touches that location, so the greater biome. And I read a paper by Zach Bush who has said, our gut microbiome is a direct extension of the nature that we touch. And think about that. That is so true. So I want to thank you all for your time today and for allowing me to speak. I am so grateful for the opportunity to work in an industry that's actually changing the world and other galaxies. Right now, you know that too. But I'm also grateful for the learning experience over the past 21 years And to be honest, I'm even more excited to see what the young scientists that are doing, that are coming after me. I'm at the end of my career, right? We got these new scientists that are coming out. So think people at Microbial Insights and other companies, what we're going to do and see them do to change the world. So as Mark Twain said, knowledge becomes wisdom only after we put it to good use. And that's what I want to see us do, is take this knowledge, make it practical. The future's bright. We just have to learn about the microbes in our bodies, in our cities, in our country, in our planet, and eventually out to the galaxy. So thinking about all the great work that you guys are doing and the pride of our industry and the change and the impact that we're having, I just want to leave you with a quote from one of my favorite movies and hope that the science continues, the research continues, and that you go to infinity and beyond.